Yes,
Yes, yes.
yes. Okay, cool. So, as I mentioned, we are coming from a citizen science program, and it's really good to hear that there's some of you who already have a lot of experience with citizen science, and also some who don't. So we have been working with schools, with school teachers who have been participating in different research activities in South America. And for this project, we have teamed up with our colleagues from the Smithsonian Environmental Research Center and from other networks, scientific networks all around the world, and we want to investigate together these ocean travelers. And in this presentation today, we have four parts. First, we will introduce the problem very briefly. Then we will look carefully at the sampling protocol. And then we have some interesting things, some interactive exercises, a virtual sampling of litter on the beach, where we need you all to participate. And then we have a very brief recap at the end. So why are we actually interested in this problem of the organisms of these ocean travelers? When we go to the beach and look at the litter on the beach, it basically can come from two main sources. It can come from the sea, where it has been moved in one way or the other by the ocean currents that move along the surfaces of our oceans. But a lot of the litter on the beach also comes from other sources, from the beach visitors. That can be tourists, that can be fisheries, that can be aquaculture or other activities that happen on the beach. But if the litter comes from the sea and has been traveling in the ocean with the ocean currents, then this litter also has been colonized by organisms. Many organisms look for some place to live on, to settle on. And we are interested in those organisms because with this floating litter, they can potentially come from far away. And so we want to know where this happens and who is coming on this litter, who is arriving on our shores on this litter. So the questions in this project that we all want to do together are: first, how much of the litter on our beach is arriving with the ocean currents? And then, of course, who are the organisms who are arriving on this floating litter? And since we are people from all over the world who are participating in this project, we are also trying to answer the question how the amounts of litter are varying, that the litter that is coming with the ocean currents, how these amounts are varying on the beaches around the world. And just to give you a very quick idea about the organisms, there can be many different organisms growing on this litter. There can be animals, there can be seaweeds, algae, and there can also be animals like these here on the left side, who are highly specialized to live on anything that is floating at the sea surface. These animals have been around for millions of years, and nowadays they actually have found this new floating object, the floating litter, and they also get transported on this floating litter. So in this sampling that we will be looking at now in more detail, we are focusing on the first two questions that we have seen: how much of the litter on our beach has come with the ocean currents, and then who are the organisms that are being transported on this floating litter. So here on this slide, we will just look very briefly at let me we will look very briefly at the main steps. So the first step is to gather your materials, to coordinate the people who will participate with you, your team, gather your team. And we recommend that the team that does the sampling could be 3 people or 4 people. It can also be done with 2 people. Danny and we recently did a sampling which we just did by ourselves, and that can also be done. The ideal number, I think, actually is 3 people, but it can also be 4 people. So when we come to the beach, we collect the litter, then we classify it in the litter types, we measure its buoyancy, so whether it floats or whether it sinks, we check whether it has any organisms growing on it, then we spread this litter on a sheet on the ground where we organize it, and we will see that in more detail in a minute. Of course, we count the litter in the different categories and we record the data. We then, very important, we take a picture from above of the litter, of all the litter that is spread out in the different categories. We will see that in a minute. And then we are basically ready with the first question, and we can advance towards the second question. Those items that do float and that have organisms growing on it, that have these ocean travelers, we then take more detailed pictures of these organisms. And we, of course, also record the data. And at the end, the only thing that is left, then send the data and the images to the coordinating team that will do the data validation and also the evaluation of the information. What do we need? We need very basic things like a trash bag. We need a clipboard, a ruler to measure things is very important. A phone with a camera. You can also be, any camera that we have nowadays works for it. Have very good cameras. I mean, any phone. But if you have a digital camera, it would also work. We need a bucket where we then can determine whether the things that we collected float or not. We need a sheet, a fabric where we can organize the litter on the ground, and of course we need to print the data tables and the labels, which we also need to bring to the beach. And what is an important task for you, you need to register the beach which you would like to sample. You need to decide the day when you want to go out. Of course, you need to organize your team, your 3 maybe collaborators or 2 collaborators. You need to gather all the materials that you need. And of course, before you go out finally, you should check how the weather is coming along, especially if you are in areas where you have typhoons or rainstorms or hurricanes. You want to be sure that you don't go to the beach on the wrong day. So that is before you go out. And then when you come to the beach, the first thing that you should do is take a panoramic photograph of the beach, which gives us an idea how the beach looks like. And then you also can set up what we call the beach laboratory, where we're going to process the litter that we collect. And that should ideally be a place that is protected, that has a little bit of shade, that is protected from the wind, and where in generally you can be comfortable, because that is where we will spend a lot of time to later, after we collected the litter, to analyze it and organize it on this sheet where you can already see here. We organize the litter in the main litter categories: plastic, metal, paper, glass, fabric, processed wood, and non-identifiables, things that we don't know really what they are, but they are litter. And then here in the columns you can see we organize it depending on whether they have organisms growing on it or not, and then whether the litter floats or whether it doesn't float. So that is very important, and all the litter will be organized on this sheet. Of course, make sure that this sheet doesn't fly away if there's a little bit of wind. If there's a lot of wind, then maybe you want to try to find a closed room where you are really protected from the wind. But if there's no wind or just a little blow, then you can do it easily on the beach. And this, I always say, is almost the most important slide of all, because this shows the area where we're going to collect the litter. And we collect the litter here in this area of the beach, which is above the last high tide line, so where the last tide dropped material, which can be seaweeds or litter itself, above that line and all the way up to where the dunes are beginning, where the terrestrial vegetation starts, can also be in palm trees or other vegetation. So all this area of open sand here between the last high tide line and the dunes or the beach promenade, that is where we're going to collect the litter. And we collect the litter randomly. That is very important, and we recommend you to collect about 100 litter items. It can be 120 or 130, that is not a problem, but it shouldn't be 300, because if you collect 300 or 400 items, then you have to process all these litter items, measure for all of them whether they float or not, and whether they have organisms or not. And I have been out a couple of months ago with some volunteers who did collect like 300 items, and at the end, after we spent like 1.5 hours processing all of this litter, they were actually quite tired and thought, oh my gosh, if we wouldn't have collected that much litter, it would have been much easier. So we recommend about 100. Can be 120, 130, that's not a problem, but don't collect 300. When you start collecting, record the time with your phone or whatever watch you have on your arm. And when you stop collecting, when you have your 100 items, or 120 items or 30 items, then you check the time. How much time did you collect together with your team? If your beach is very clean and after 30 minutes you still have not collected 100 items, you may stop. You can also, if you are still full of energy, collect a little bit more, 10, 20 more minutes, but you don't have to stay on the beach the whole day to gather the 100 items. If after 30 minutes you think this is it and we can't find much more, then you may stop. And if you found 40 items after 30 minutes, well, those are the 40 items that you will process. We also focus here on the macro litter, which we call macro litter. So that is a bottle cap or larger. So everything that is a bottle cap or larger we collect, and things that we don't collect is cigarette butts, face masks, food remains or personal hygiene items, for the obvious reasons, because we don't want to expose ourselves to these items. And as we said, we collect the litter randomly. So for example, here on this beach, we walk around and everything that crosses our path, we collect this and put it in our bag. And while we do this, we count a little bit and we talk to the other people. And when we all feel now we have about 100, maybe we collect a little bit more, and then we stop because that is enough for our sample. And for example, on a beach like this, it's very important that you take the time, because on a beach like this, for example, you will collect the 100 items in 2 minutes. There's so much litter here on the beach that it's not going to take a lot of time. Hopefully, your beach is not looking like this. And we have already received from some participants information and data that indicate that their beach did not have a lot of litter. But many beaches, people within 10-15 minutes collected their 100 items. And so that's also an indicator that we do have a problem. Okay, so we collect the litter on the surface and we don't dig up litter, and of course litter that we can't carry, that is too big, we also don't move it. We don't bring it to our lab. And once we come with all the litter to our beach laboratory, we separate the litter into the main categories. And the main categories that we distinguish here are plastics, metal, paper, glass, fabrics, processed wood, and others that we don't know really what it is. And we do that to the side of our sheet, not on this sheet, because they go on this sheet only later, after we know whether they float and whether they do have epibionts or not. So once we have organized the litter here into these main categories, then we actually start to determine whether our litter item is floating or not. So we dump it in the bucket and we check, we remember the result, then we inspect the litter carefully whether it has these epibionts or not. And for example, in this example, we see that our plastic bottle here, it did float and it did not have any epibionts, any organisms. So then we take our bottle, and here on our sheet on the ground, we put the bottle where it corresponds. So we had a plastic bottle, it did not have any epibionts, and it floated, so this plastic bottle goes here together with all these other items that have no epibionts and that floated. And so on this sheet we organize all the litter in the corresponding categories, whether it's plastic or metal, paper, glass, or whatever it is, and then whether they have epibionts here, like here on the right side, or whether they don't have epibionts, and whether they float or don't float. And once we have done all this, basically we count, once all our litter items are here on this sheet on the ground, we count them in each category and we put the data in our data table here. And if you paid attention, you can already see that this data table is organized exactly in the same way as the sheet, the litter on the ground. Here we have the plastics, metal, paper, glass, fabric, processed wood, others that we don't know what it is, non-identifiable. And then here on the left side we have the items without epibionts, and on the right side we have the items with epibionts. So you can see here on the ground we organize it in the same way, right? On the left side without epibionts, on the right side with epibionts. And so when we count each category, then it's also easy to transfer these data here in this data table. And you can see here an example from some of the teams that have already collected here in Guatemala. We see here they have put the name of the beach. Of course, that is very important. The coordinator, who is the person, the responsible person, which would be you, who is in contact with us, the name of the team. And here, this is also very important. Remember that the time that they have searched. So in this case, for example, they searched for 10 minutes and all these items they collected in 10 minutes. And with this data table, actually, we can now already answer our first research question, and that is how much of our litter actually has come with the marine currents. And we assume that the litter that does have epibionts and that does float is the litter that has come with the ocean currents, because if it doesn't float, it can't have been transported with the ocean currents. And if it doesn't have any marine organisms growing on it, then also it didn't pass a lot of time in the sea, and it was not colonized, probably deposited on the beach by beach visitors or by other sources. So here we can now respond our first question, and we are almost ready. With one exception, we now need to take an overhead picture of all this litter as it is organized on the ground here, because this picture will be very important for our team to validate the information. Because our team will then count the litter here from the picture in each category and compare it with the data table. So for example, we have seen here there is 3 plastics that float and have epibionts and 1 that doesn't float. If we go back here, we can see here is the 3 plastics that do have organisms and that do float, 1 that doesn't float. And the other thing that you can see here, for example, in this category, the plastic without, or the plastics without epibionts and that did float, there's a lot of items here, and so it's very important that you organize it so that our team later can recount this from the picture. And if there's a lot of litter, you can also make small groups here of 10 items that you put together, and where you put a little label that we have that has the number 10, and so that tells us, oh, here is 10, here is 10, here is 10, and here's 10, so that's already 40, and then they only need to recount these items, so that makes it easier also for our validation team. Because then we need to go back to this table, right? And if it says 66, if the team here from the picture counts 64, that is okay. But if it here says in the table 66 and on the picture we only count 30, then we have a problem. So this picture is very important, and you can see here also this team, they did this very well. They gave more space to the plastic, which is what we usually have most, and the metal, paper, glass, fabrics, other things here on the bottom, they gave it a little bit less space because, as you can also see, there is fewer items. So this is the first question, and now we move forward to the second question where we are interested in the organisms, and here we focus on the litter that can travel with the ocean travelers, the litter that does have epibionts and that does float. So here, and of those items we then take more detailed pictures, and if we have many of those items with epibionts, we select the 10 that have most items. If we have only 4, like in this case, we have here 3 plastic items that floated and has epibionts, and there's 1 other item down here. If we have only 4, well, then we can only take detailed pictures of 4. And in some cases, actually, there were groups that didn't find any litter with epibionts. So that can also happen, and that is also an important result if that happens. But if you do have these objects that do have epibionts and that do float, then we take more detailed pictures of those, and those pictures are very simple in a way, very straightforward. So each item that we take a picture, we have its own label. Somebody has a microphone on. Can you check? I can't switch it off from here. Could you check that your microphone is switched off? That would be great. We are almost there. We are almost ready. Give me a few more minutes. My cable here is. I'm getting entangled, just like some organisms get entangled in the litter. So the important. Now we take these pictures of the objects that do float and have organisms. Of each object, we take 3 pictures. We have the label for each object and the ruler. So the first picture is the side A, where we have most of the epibionts. Then we turn the litter object around. The other side, that is the side B, and then the third picture is the picture C, where we take a more detailed picture of the epibionts that we have. And you can see on this picture the ruler is very important. On all pictures here we need to have the ruler, because the ruler will help us to measure the litter object and also to measure the size of these organisms, because the larger they are, the more time this litter item has spent at sea, and so from farther away it could have come. So it's very important to have the ruler in each picture and of course the label. And then the only thing that we need to do for each object, we fill this out in this table here. Each object will be recorded in this table, so like our colleagues did here. So they have all the different objects here in this table, and with the help of this table we can then match the litter objects that each group is sending, and we can also respond with the help of the pictures of the litter object and the epibionts. We can also respond our second research question: who are these organisms, who are the species that are being transported on this floating marine litter? And now we are almost ready. We only need to send the pictures of the data table of this sheet with all the litter to the team. This is the first question here. And then for the second question, we have also the data table with the litter objects, the litter items, and the 3 pictures for each litter item that we need to send, and also not forgetting the panoramic photograph. And since we are many people from many different countries around the world, together we can also respond the third question: how these amounts of litter that is coming with the ocean currents is varying on the beaches around the world. And that is all the sampling there is. That is what we have to do on the beach to do this sampling and to participate in this project of the ocean travelers. And I wonder whether there is any questions about this. Does that sound clear, and does it sound doable for you? Hello, Jessica. I see your note in the chat. Anybody has any questions? Yes, Ruth.
I've got a question. Of a lot of the litter that we have on the beach is sort of fishing gear, sort of old fish netting. I didn't see that in any of your category. Do you recognize that? Is that in plastic, or how do you deal with it?
Yes, that is a very good question, and that indeed, if we go back, let me do this here. So let me go back to this. Where are we? So we can go maybe to this slide here. Yeah, this is a good one, I think. So here you can see these are the categories that we distinguish, and Ruth is asking, yeah, where's the category for fisheries litter? There is indeed no category for fishing litter, and those of you who have participated in previous projects, for example, I suspect many of you have participated in the International Coastal Cleanup, and then you know already that they distinguish a lot of different categories, including, for example, even different types of fishing litter. But we have decided to keep it as simple as possible in this case. So the fishing litter would be, depending on what it is primarily made of, whether it's plastic or metal or other things, most of it, like for example buoys or ropes or rests of nets, would be then in the plastic category. We do recognize, of course, that we lose some information, but on the other hand, we wanted to keep it as simple as possible so that as many people as possible can participate. And also in this particular research, also we are not interested so much in pinpointing who was responsible for this litter, but we want to find out on which coasts we do receive a lot of floating litter that is coming from elsewhere, and what organisms are arriving on our shores on this litter. So that's the main goal of this research, and not so much say, oh, this most of our litter is coming from the fisheries or from other sources. So we don't distinguish specifically the fisheries litter. Are there other questions?
Yes.
Yes, Ngozi.
Okay. How many times are we going to do the sampling?
Yes. Oh, sorry, I did interrupt you.
I said how many times
Yes.
and how soon?
Yes, how soon? The sooner the better. So this project will last until the end of the year. And also, as always, now, for example, you have it all very fresh, what needs to be done. So if you wait another month, then maybe you will forget a lot of things. At least that's what would happen to me. So if it would be me, I would try to do it maybe during the next 2 weeks before I forget some of these things. And that also happens to me. Sometimes our team needs to remind me, oh no, this goes in this category. So that would be that recommendation. And with respect to how often to do it, this is a really good question. We know, of course, that in many places of the world, the amount and whether the litter is coming from the ocean or not is varying over the year, depending on the season. For example, we know in many tropical regions where we have the dry season, the wind is blowing from one direction during the dry season, and it's coming from totally different direction during the wet season. And that would, of course, affect the amounts of litter that come from the sea. But in this project, we are not looking at the seasonal variation. We just are interested in getting a first idea what litter is arriving from the sea and how does this vary on a large geographical scale, basically around the world. But your question is really good. In the future, we are thinking, we have been discussing that this question that you ask, other people have asked this before, and of course, we have also asked ourselves that. But for the moment, it's only one sampling, only one time sampling at this time. Yes. Any other questions?
I have a question.
Yes.
So we work both here in San Diego, but also in Mexico. Can we sample once here in San Diego and once in Nayarit in Mexico?
Oh yes, of course. That would be wonderful. Yes, I mean,
Okay.
fantastic. Yes.
Thank
And
you.
if somebody of you, I mean, we are talking to you because we think that you will probably sample the beach right in front of your house or of your lab where you are living and working. But if you travel to other places around the world and you see a beach and you have some time on your hand and you are curious yourself what's happening on this beach, you are more than welcome to also do it elsewhere. Any other questions?
I've got another question,
Yes.
Martin. So just to see, about the floatability of the litter. In your experience, is it really clear cut whether something is floating or not?
Okay, very good question. And if there is no other urgent question, I would actually say this is the perfect question to jump into the virtual training, because that question in the virtual sampling that we have, because that question actually will be responded, I hope, I think, in the virtual sampling that we do. So if nobody else has another urgent question, and I don't see anybody, then I would say let's move on to the virtual sampling, which we actually designed during the pandemic usually, and in the past, since we have been working mostly in Latin America, we have done the training sessions in person. So we have gathered people together and we have done a sampling together on the beach to do the training, which of course is much more fun. Now during the pandemic, that was pretty much impossible. And also now, since we are spread so far around the world, it's also almost impossible. Nobody has the money to get us all together, which of course would be a lot of fun, but we can't do it. So we have this virtual sampling, and we'll jump into this virtual sampling now. I will share again my screen. And so you remember the first thing when we come to the beach, we need to identify our sampling area. And so here we see an overview picture that is taken with a drone. Somebody took this with a drone from above of a beach. And so we need to identify the lower and the upper limit of our beach. So the last high tide line, where would we put that? What do you think? This line, should it be somewhere here, or should it be higher up? Should it be somewhere here, or should it be lower? What do you think? And just open your mic and say higher, lower, whatever you think.
Lower, lower, lower.
Lower,
Lower.
lower, 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 lower. Much
Lower.
lower?
Here.
Aha. Yes.
Yes,
Stop.
okay, perfect, perfect. So, okay, this is, you know, where the sand is still clearly wet, where the last waves washed the sand and left the last stuff that came. So this would be the last high tide line. And where is the upper limit of our beach? Where do we need to put this line?
Higher.
Higher?
Higher. Higher. Yeah, about there.
About
Yes.
there, yes, exactly. So about here, so this is then in between these two lines. This is our sampling area where we would collect the beach on the litter on this beach. Okay, so now we know where we are collecting, and the question now is, what do we collect? So here's our sampling bag, our litter bag, and how about, for example, this magazine here? Do we collect this, yes or no?
Yes.
Yes, definitely. How about this cookie wrapper? This is kind of like you cannot recognize. I couldn't recognize it first, and somebody had to explain it to me, but it is a cookie wrapper. Should
No.
we
You,
no?
No.
Yes or no?
No.
yes.
Yeah, we should collect it because it's also litter, so we also collect it. How about this plastic cup? Would we collect this?
Yes.
Yes.
Yes. Okay. How about this bottle?
Definitely.
Yes.
Yeah. How about this can?
Yeah.
Mm,
Yes.
Yes. How about this bottle cap? I make it a little bit larger since I have a magic touch.
Yeah.
So
Yes.
yes, we collect this. How about this cigarette butts?
No.
No.
No. How about this bottle cap that does have something growing on it?
Yes.
Definitely.
Yes, definitely. How about this wooden ice cream pallet?
Um,
Y-
well,
y-
it's processed timber, so it was the category.
Yeah, it's processed wood in a way. So yes, we would collect this, and of course the same with this one. Oops, I didn't get this. How about the cardboard roll?
Yes. Yes.
Yes, excellent. How about this lemon?
No.
No.
No, that we don't collect, just like the banana peel. How about this big buoy? Do we collect
Yes.
that?
Yes.
Yes, if we can easily carry it, we also collect that. How about this can?
Yes.
Yes.
Definitely.
Yes, how about those pens here? They still seem to be quite good.
Yes.
Yeah.
Yes.
Okay, well, first we look whether there is somebody on the beach who these pens might belong to. But if there's nobody, then we do collect it. Okay, so you get the idea, of course, the plastic bag as well, the
Yes.
plastic bottle.
Mm.
How about this piece of wood here?
Yes.
No. It's driftwood, huh?
Yes, so this is natural wood, some branch broken off from a tree, and so it's natural litter, natural debris. We don't collect it. We only collect anthropogenic, like human
Okay.
debris. So, for example, this ice cream wrapper, of course, we would collect. And okay, you get the idea. We don't need to continue this. Let's then jump to the next thing. We have.
I do
Oh,
have one question.
yes, please, Jessica.
If we do find the driftwood with Epibionts on it, would we take a picture anyway?
You may, but we will not consider it. Is a very good question, and of course, it's also an important research question. But in this project, we only focus on the anthropogenic litter. But of course, if you take a picture and share that with us, we would be also happy to receive it and share it with our social networks, for example. So if you have an exciting find, and think there's an interesting picture, please also share that with us. But in our sampling, we are not including it.
Okay.
Okay, cool. So we go then to our beach laboratory, where we have already established this sheet on the ground, and we have already sorted the litter into the main categories. So we have the plastic here, we have the metal down here, we have the papers here, we have the glass here, we have the fabric here, and we have the processed wood. And so now we are ready to move forward and determine the buoyancy and whether there's organisms. So how about this plastic bag? What do you think? Does it float? Yes or no?
Yes.
Yes, maybe. And as always, there's only one way to find out. You need to put it in the bucket, dump it in the water, and it does float, and it doesn't have any organisms growing on it. So we move it over to our sheet on the ground here, and this plastic bag then goes here into this category, plastic without epibionts, and it did float. And then the next plastic bag, well, we would think it also would float, but we test it out, and you can see here, so this goes already in the direction of Ruth's question. It doesn't float as nicely as the others, so we wait a little bit, but it doesn't sink to the ground. It remains suspended, and so we also consider that as floating, and it's also without epibionts, so it goes right next to our other plastic bag here into this category. So how about the ice cream wrapper? What do you think? Does it float? Yes or no?
Can you
Well,
check?
maybe,
I think
maybe
yes.
not. There's only one way to find out. Put it in the bucket, it floated, and it doesn't have any epibionts, so it also goes here into this category. How about this plastic cup here? What do you
Yes.
think? Probably yes, and indeed it does float, and so we move it here, and so there it is, there it goes, and the same we suspect, but we have to try it. Happens also with the other one. So yes, same thing, and also clean, also no organisms, so it goes in the same category here. And so you can see already your litter is kind of like getting into different categories. How about the bottle cap? And this is actually a good reminder. You know, so you may say, oh, why did you do? We already know that it doesn't float. Well, you know, a couple of weeks ago I was out with some volunteers and they had a bunch of bottle caps, and I said, oh, they're all gonna float. But to my surprise, actually, there were 2 bottle caps among them that did not float. And so that is a really good reminder that you need to test it for all your items. This one did float, and so it also goes here into our category of litter without epibionts and plastic, floating plastic. So how about this one here? Well, does it float? Yes or no?
Probably.
Yeah, probably. So let's try out. And yes, it did float, but this one has organisms growing on it. So we take it, and now we need to move it into the category with epibionts and plastic with epibionts that did float. So here it goes. And then we move to this plastic bottle that is empty, but it does have a cap. Does it float, yes or no?
I think yes.
Everybody else agrees?
Yes.
Yeah, most
Yes.
likely. It's full of air, so it floats, and we can see it has also a bunch of epibionts. So it goes here into our category with epibionts and floating. Now, what do we do with the next bottle? This bottle doesn't have a bottle cap, but it's empty. So what do we do with this one? Does it float, yes or no? What do you think?
I think so. It's still light enough.
Yeah, it could be, but it's very important. So we take each liter the way it comes into our hands. And in this case, what we do, like with all the litter, we hold it underwater. And if there is air, if we see that air is coming out, we keep it underwater until no more air comes out. And then we let it go. And if we do that, actually many plastic bottles, not all, so we have to do the test, but many plastic bottles, especially the PET bottles, they will sink. So, so the important thing, hold it and make sure no air is coming out. As long as air is coming out of your litter object, you need to keep it submerged until no more air comes out, and then you let it go. So this bottle then actually, it had no epibionts, but it did sink. So it's the first plastic item that goes into this category that sinks. And how about this lighter? What do you think? Does it
I
float?
think it's no, I don't think so.
Really? Do we have any smokers here? Or anybody who has found some lighters on the beach?
Yeah, you do.
Yeah, so they usually float, unless, of course, they are broken. And when you submerge it, air is coming out, then you keep it. And then actually, I don't know. But I think actually even the broken lighters still float. But of course, the lighter, we do know they do have some metal, but most of the lighter is actually plastic. So more than 50% is plastic. So this lighter actually then goes onto our sheet here where it is. We did say it has no epibionts and it floated, so it goes here. And then how about the cookie wrapper?
Yeah, it's light.
Yeah, but you need to find out, and you need to make the test, and it did actually sink. So it goes together with the bottle here, with the plastic bottle. There's nothing growing on it. And how about the processed wood here, this little ice cream pellet?
You put it in the water and see if it sinks.
Yeah, okay. So since it's wood, a lot of wood floats, but not all wood. There is a bunch of different wood types that do not float, but this one did float and it didn't have any organisms growing on it. So we move it here into this category, processed wood without epibionts, and it floated. And we do the same thing here. Also, it did float as well. So we move it here together with the other one. Yeah, and you can see now, actually, you get the idea. The paper, does it float, yes or no? We don't know. Test, okay, this one did not seem to float, so paper, we move it here. The paper, no epibionts. This looks very clean. Somebody left it on the beach maybe a day before. And the same here. I will not move it. Let's move forward. I'll just do 2 more tests with you. How about this bottle? What do you think? Does it float? Does it sink? It has a lid on it, and it's still
Yeah,
full
and it
of,
looks full.
yes, it's full of beer.
Yeah, well, then it
Well,
won't float,
for me
huh?
it's early in the morning, but for some of you maybe it's already time for a beer. I don't know. So, but it does it float? Yes or no? If we don't know, only one way to find out. No epibionts, so we move it here to the glass, and it did float. We move it so that it fits in, that it can be easily counted. And then how about this bottle?
Probably not, because it doesn't have, or the air can sort of
Yeah,
get out, and then
yes,
glass is probably heavier than water.
yes. It's broken, yes, and so same with as we did with the plastic bottle. So we hold it underwater until no more air comes out. We let it go, and then it will sink, and so we need to move it. Okay, you get the idea. Okay, let's move forward. So, okay, we do this. Then we have all our litter here on our sheet. We fill out the table. Don't forget to put the name of the beach, the coordinator, which would be you, the name of your team, and how much time you actually collected. That is very important because it gives us an indication of how much litter there was on your beach. Okay, and once you have filled it all out, we are ready to respond our first question. But don't forget, take this overhead picture of all the litter spread out on the sheet in the different categories. See here, organize the litter so that it can be easily counted from the picture, because this picture is very important. If we don't have this picture, it's basically as if there was no sampling. So this picture is very important for the validation of the data. And then, as we have said before, we take the detailed pictures of those items that do have epibionts and that did float here. This can actually, I should have done this test as well. This can is closed, so there's also still the Coke inside, and then it does float just like the beer bottle that we saw. So now we need to take the pictures, the detailed pictures, and remember the first picture here, we take the picture of the side that does have the epibionts, and we need to make sure that the ruler is always on the picture. Then we turn the bottle over and we just mark in the tick box here down there, we mark that this is the picture B of the first object, and then we take a close-up here of the epibionts, right? And oops, nope, I didn't want to move the ruler. I wanted to move the bottle so that, oops, I can't move it. Oh, I did something wrong. Let's do it this way then. So okay, and then we mark here that this is the picture C, and we make sure that on our picture this letter and the number is there with the ruler and with the close-up of our object. And so we do this for all our items. So each object, each litter item that does float and have epibionts, we have 3 pictures, A, B, and C. And then, of course, we need to fill out this data table where we put the category. It was a bottle, right, and it was 21 centimeters, and the other one was also plastic, and that was the bottle cap, and we already know the bottle cap measures 2.5 centimeter, and the last one actually was a metal, and it was a can, and it's like 12 centimeters in height. And so we are ready basically now. The only thing that is left, we have the pictures of the litter on the ground, of the data table, the first data table, and that is for the first question. Then we have the table of our litter object that did float and did have organisms, and of course all the pictures, and we send all this together to our coordinating team. And yes, we don't forget this. And then basically we are ready, and we can lean back and say we have done one sampling, one beach, and contributed to this project of the ocean travelers, and put our beach on the map. Do you have any questions now? Ruth, and yes, Guka, 1 second. Ruth, did this now respond your question about sometimes you don't really know?
No, it's perfect, really. So
Okay.
I totally get it. Thanks.
Okay, Guka.
Yeah, just one small question. What was again the photo A and B? I didn't get it right. So it was the A and B in the photos
Oh,
of the
okay,
item.
okay.
So
Yeah.
you check the A and the B.
Yes, okay, sorry. Let's go back then. Let me go back and share the screen, and let's go back. So let's look at this one. This is, let's just focus on our object number 1. So the first picture, the A picture, is the side where we have most epibionts. Then basically we take this picture of the picture A, then we turn our item around, look at it from the other side, because there may be other organisms on the other side. Often in this example here, there's nothing on the other side. But often you have some organisms on one side, and on the other side there may be other organisms. So that is then the picture B, and you can use the same label here. Just mark the tick box B, and then the third picture is the close-up of the epibionts, right? And there actually, since the whole label doesn't fit, we just said make a small label that just has the number and the letter C. And actually yesterday in a training session, somebody asked a very good question, because as you can see here on this bottle, there is one organism here. We have this close-up of this organism, but there's a different organism, a little seaweed here. And somebody asked me, oh, so should I take 2 pictures, 2 sea pictures? And actually my answer is yes, you should. So if you have some, and if you can clearly recognize that there's some species in one corner, and when you do the close-up you see those, but you don't see the different species on the other side of your object, take another sea picture. So you may, depending on if you have many different species of organisms, you may have 2 or 3 sea pictures. But the important thing is A, B is the whole object, and C is the close-up of the object. Does that respond to the question? And hopefully not introduce any confusion now. Any other question? Okay. No other questions.